are going to discuss about new topic. Today topic is chromatin. What is chromatin? So first of all, who gave the word chromatin? This word it was given by Fleming. Very important questions. Remember, 1 mark questions. The term word chromatin it was given by Fleming. The second one, what is the meaning of chromatin? Chromatin means colored substance, colored material. Means any material which can be easily stained with the help of some dyes. When we can be shown a detailed structure, when we can study the microscopic structure of this component which is called chromatin, so it can be easily stained. This chromatin it is found in non-dividing state of cell. When we study the cell cycle, cell division in the next chapter, the one word which is given here, this is called I phase. The detailed structures of chromatin it can be seen in non-dividing state of cell. That non-dividing state is called I interphase. I stand for interphase. Suppose this is the mother cell. After division, it will give the two daughter cells. Depends upon whether it will undergo mitosis and meiosis. In meiosis, 4 cells are formed. In mitosis, 2 daughter cells are formed. So this is our primary mother cell. When we will study the cell cycle, cell division in detail, the simple diagram is given. Basically, there are two phases. This is very long phase, which is called non-dividing phase. This phase is called I phase. I stand for interphase. This is further divided into the 3 subphases: G1, S, as well as G2. So means this the bigger portion of cell division, which is called I phase. In this, what we will see? We see this thin fiber-like filament, which is called a chromatin. So these fine filament-like structure, which can be seen within the interphase, this is called chromatin. When this is highly coiled, compact, condensed, it can give the further formation of rod-like structure which is thick. This rod-like structure is called a chromosome. It means chromosome. They are made up of well-coiled, condensed form of this fiber. This fiber name is called chromatin fiber. After that, what will be happen? When we study the chemical compositions, the chemical composition of chromatin, it is made up of linear double-stranded DNA plus some amount of RNA is also present. Some enzyme are present, DNA polymerase, RNA polymerase. Instead of this one, some metals ion are also present in this one. The best one is it also consists of some protein. Here there are 2 types of proteins are present. One is called histone, another is called non-histone. Again, very important questions. This histone protein is basic in nature. Basic in nature. Another protein which is called non-histone protein. These may be acidic, these may be neutral. 1 mark question that may be setups. When we study in 12th the packaging of DNA in nucleus, the one what is given which is called a nucleosome. This nucleosome it is nothing in which central core particle which is called protein. This protein are wrapped by DNA molecule. This structure is called a nucleosome. Means DNA plasmid protein particle which is called histone is called a nucleosome. These histones are 8 in number. That's why the word is given as octamer. This histone is called structural components. Very important questions. And the remaining portion of protein which is called non-histone protein, this is called functional protein. The functional components which is called functional protein. So this is called structure because basic structure that is made up of they have these protein which is called a histone, whose nature is called basic in nature. Very important questions from next point of exam. If you say this is called, what is the nature of histone? Basic natures. What is the charge on the histone? They will carry the positive charge. What will be the charge on DNA? DNA having negative charge. That's why there is a more attraction between negative charge particle. The name of negative charge particle is called DNA, which is called genetic material, and the positive particles is called protein. The protein here name is called histone, and they have the basic in a nature. So these are the two type of protein which are also present here: histone plus non-histone. So that we move next. How many types of chromatin? Basically, there are two types of chromatin. Look here. One type is called euchromatin. Second is called a heterochromatin. So there are some difference which are given here. How we can make the difference between euchromatin as well as heterochromatin. This euchromatin, this is called true. Heterochromatin, which consists of the different components in it. So let me study. Euchromatin, it is thin in nature. This is called thick. This is uncoiled. This is called coiled. That's why this is the extended form of chromatin fiber. Here is the compact form of a chromatin fibers. Next we can, the major portion of chromatin is belong to the euchromatin. That's why it is called bulk of chromatin. But the heterochromatin, it is present in the small amount, small fraction. So it is having the little amount. So it can be easily stained, but the stain is light in color. But it is compact, that's why it is a dark stain. Another difference, it is used in transcription. This part is not used in transcription. That's why it is not. It is involved in transcription. It is transcriptively active. This is called inactive. Here the gene are present. They are in the active form. Here the gene which is present here, they are known as inactive. Means they don't express. They don't give any kind of phenotypic traits which is formed here. These are some difference which is given here. Euchromatin versus heterochromatin. So let me study the detailed structure of chromosomes. Suppose this is a chromosome which is belong to a stage. This stage is called metaphase because from here to here, what will we call it? This is called I phase. This is called interphase. This interphase is called non-dividing state. This is called non-dividing. Next, that is called M phase. This is called mitosis or mitotic phase. This is called dividing phase. And the first phase of this mitotic M phase is called P. P stands for prophase. The second one is called metaphase as the name given here. The next one is called anaphase, A for anaphase, and the fourth one is called T for telophase. So suppose this is the chromosome. It is nothing. It is a highly coiled, compact form of chromatin fiber which is look like as a rod. So this thick rod-like structure which is formed by the condensation of the chromatin fiber is called a chromosome. A single chromosome having the two arms, upper arms and lower arm. These arms may be equal, these may be unequal depending upon the location as well as position of this point which is called centromere. Here these arms are not equal, these are unequal. This is the short arm, this is the long arm which is mentioned here. What is this? This is the chromatin filaments. This chromatin filament which is present in the condensed form and which contain the DNA double helix, this is called chrominal. This is the disc-like structure which is called primary construction. It is also called kinetour. At the kinetour, the central point which is called centromere. From this point, these arms are attached within a single chromosome. This is called upper arm, this is called lower. Here the size is unequal. This is the short arm, this is called long arm. Instead of primary construction, more than one construction may be present. The another word which is called secondary construction. If the secondary construction it is present at the tip region and the small portion of chromosome is remaining behind, this is called set. Then you can say satellite. such kind of chromosome in which secondary construction is present. This is called set chromosome. The one word which is given in your NCERT, what is satellite? This satellite also behave like a marker. That portion of chromosome beyond the secondary construction is called satellite. Such chromosome which having the secondary construction is called set chromosome. So this is the simple diagram of a chromosome. This chromosome is taken from the stage which is called metaphase, or this metaphase stage is the best stage, you can say phase of cell division, in which we can see the best visual structure of a chromosome. So out of 4 phases, which one is choose? Metaphase. Again, 1 marks question that may be set up from here. Out of 4 phases, which one is the most stable phase to observe the best visual picture of chromosome? So this is called metaphase. This is called anaphase. So depending upon the position of it, the central point at which these chromatid are attached, these arms are attached, upper and lower. How many type of chromosome? In your book, there are 4 type of chromosome are given. Look at the blackboard. Depending upon the position of the centromere, we can be classified chromosome into the 4 category. Here, 1. This is the first one. The name of first one is given is metacentric. What are metacentric chromosome? Those chromosome in which the position of center is at middle. It means these arms are equal. That's why this is called metacentric. What is the location of centromere? At the midpoint. It means the size, the length of these 2 arms are equal. So that's why such kind of category of chromosome is called metacentric. Next chromosome is called submetacentric. As the picture is compared, when the picture are compared, it means the position of centromere is not at the midpoint. It is away from mid. It is away from middle. So that's why the word is given submetacentric chromosome. It means the position of centromere is slightly away from center, slightly away from the middle. So that's why it is called submetacentric chromosome. The third type is called acrocentric. It means in which position of centromere is far away from a center. It means it is not at the middle. Middle से काफी दूर है. It is away from center. So that why this kind of this category of chromosome is called acrocentric. And the fourth one is called telocentric. It means the position of centromere at the terminal position, at the tip region. That's why the word is taken. It is called telocentric. It means the position of this point, which is called centromere, is at the tip or terminal end. This may be happen. It means one arm is very small and the one arm is very large. Here at the tip region, or here one is large, one is slightly small, or here one is large, another is small. So these are the 4 types of a chromosome depending upon the location. Here you can see position of a centromere. So there are 1 2 definition again which is given here. One definition which is called nucleosome, another is called chromatosome. So first of all, what is nucleosome? This concept is belong to 12, but the definition is given. Very simple definitions. Histone protein plus wrapped form of DNA is called nucleosome. Very simple definition. What are histone? These are the basic structural protein around which what is wrapped, the DNA is wrapped, histone having the positive charge, the DNA having the negative charge. So this kind of arrangement in which at the core particle which is called histone, around which something negative charge particle is wrapped. This negative charge particle is called DNA. The combination of 2 is called a nucleosome. Yeah, this structure is called nucleosome. It means one nucleosome, it is linked to the another nucleosome with the help of a linker. This linker is called DNA. Look at the figure. Suppose this is nothing, this is called rock, this is called string. Understand what are present? These are called beads, or these beads are nothing. These beads are called nucleosome. जैसे कि मोती की माला होती है और उसके ऊपर मोती अरेंज होते हैं, सेम यहाँ पर what is arrangement? Arrangement of a nucleosome. So this kind of arrangement of nucleosome, which is located on a thread-like structure, this thread-like, this is called string, and these nucleosomes are linked to each other. This is called DNA linker. So combination of nucleosome and DNA linker, this is called chromatosome. So very simple definition. Don't forget, very simple one. Combination of nucleosome plus DNA linker is called chromatosome. Or what is it? Nucleosome, the histone particle around which DNA negative charge is reacted. This arrangement is called nucleosome. So this is your topic. You can remember your topic, and these topics are given in your NCERT book. It is very important.